Please join me in welcoming to the Distinctive Voices podium, Dr. Denise Montell.
You know, a long time ago, every one of you was one single cell. A teeny tiny egg had fused with a sperm 1,000 times smaller to generate a genetically unique cell. Now, this much I'm pretty sure you already know. But then what? How did your single-celled self build a baby with almost no help from anybody else? Sure, mom provided nutrients, but you had the encrypted instructions to build every single cell in your body and put it in exactly the right place, all by yourself. Think about that. That would be like a rocket ship building, not just building itself, but building every part of itself along the way, starting with a single bolt. This is the most amazing thing you've ever done, no matter how amazing you are. So I want you to think of the problems you had to solve along the way here, right? You're now made of a few tens of trillions of cells. And so the first thing you had to do was get busy dividing, but that wasn't the hardest part. You had to make hundreds or even thousands of different kinds of cells and put each and every one into exactly the right place. Now, some of you are probably pretty good at puzzles, but can you do a 37 trillion piece puzzle without the picture on the box? Okay, but you did. So we had a lot of problems we had to solve along the way. Let's think about some of the things that we did. You actually had to roll up your own spinal cord out of a flat sheet of cells, just like is happening right here, right now. You had to fold up your gut tube and then sprout all kinds of organs off of it. You had to make muscles and bones and fat in the middle. You had to wrap yourself up in layers of protective skin. In an incredible act of cellular origami, you folded up a 4-chambered heart out of a simple tube of cells. And then you had to wire up your nervous system and connect your eyes and nose and ears and mouth and all your skin and your internal organs to your brain so you could send signals back and forth. How you did any of this was one of the greatest mysteries in all of biology until recently. So back when I was a student, we basically thought that this was an unsolvable problem. And now it's gone from seemingly unsolvable to, in many ways, solved. And not just for one organism, but for all these beautiful creatures that you see on planet Earth. So each of these organisms came from the fusion of an egg and a sperm, and then that cell had to go on to produce an entire animal. So for thousands of years, people speculated and wondered about how this might happen. But then the community of developmental biologists over the last 40 years or so, thousands of us working in labs all over the world, chipped away at our collective ignorance, gene by gene, experiment by experiment, year by year, so that now we can explain cell by cell, molecule by molecule, how this embryo builds itself. And this information brought with it gifts we did not expect or anticipate, that we could hardly have imagined. So treatments for infertility, treatments for cancer, treatments for degenerative diseases. Because we now understand so well how each cell turns into the mature cell type it's going to be, that we can do this in the dish in a lab. And we're actually on the cusp of a revolution in medicine where we can actually control the behavior of cells and get cells to remove unwanted cells like tumor cells from the body or replace damaged, diseased, injured, or degenerating cells. We're on the cusp. We're not quite there, but it's coming. And so And so what I want to tell you about tonight is I want to tell you about two stories from my lab in which we have been able to start by just studying the humble fruit fly. And by studying the cells in the humble fruit fly, we have uncovered mechanisms by which we can engineer cells to remove tumor cells from the body or prevent or treat neurodegenerative disease. So here's the star of the evening, the fruit fly. And in particular, in my lab, we're interested in the development of the ovary, the development of eggs inside of the ovary of the fruit fly. So inside of the organism, it looks like this, but if we spread it out on a microscope slide, it can look like this. And this, we love this model because of the really beautiful and simple anatomy that you see here, because of the powerful experimental tools that we can bring to bear on this organism, and because of the long history of making fundamental discoveries using this particular experimental organism. In fact, here are the 9 Nobel laureates who used Drosophila in order to uncover fundamental mechanisms of genetics, embryonic development, immunity, and animal behavior. In my lab, we're interested in how cells behave. We're interested in how they behave normally when they're building or maintaining or repairing a tissue, and then how they misbehave in diseases like cancer. So, as I mentioned, this is our organism, and I'm going to tell you about the discoveries that we've made that have allowed us to enhance a cancer immunotherapy and uncover a new strategy for preventing neurodegeneration. So to introduce the real star of the evening are actually the cells of the Drosophila ovary. So let's talk about those. Here we have the whole ovary. It's made of these repeating units, these strands, and each strand has stem cells at the very tip of it, and those stem cells divide and produce daughter cells, and the daughter cells assemble into these little ovals, and each oval grows and develops further into a mature egg. If we zoom in on one of these strands, then we can see this in more detail. Each cell has a nucleus with DNA, and that's shown in blue here. And what we have in the center of each of these growing egg chambers are 16 germ-line cells, one of which is going to be the egg, and it's growing and growing. And it's connected to 15 sibling cells that are called nurse cells. And these cells are support cells, so they get very large, and eventually they're going to donate all their contents into this growing egg to allow it to grow bigger. These germ cells are surrounded by an epithelial layer, a layer of smaller cells called follicle cells that are kind of arranged like bricks that are mortared together. But there's a small group of cells from this end of the egg chamber that as the egg chamber grows from this stage into this stage, these cells actually detach from their neighbors and squeeze in between these other cells and migrate all the way to the growing egg cell. And you can see that here in this time-lapse video of the cells as they walk away from their neighbors, squeeze in between these cells, and then ultimately reach the growing egg. And if they don't do this, the female will be completely sterile. So I was fascinated by this phenomenon. I was just curious, how does this work? What makes them go? How do they go? But the problem was that at the time that I started these studies, we couldn't even do the kind of imaging that you're seeing here. And then we didn't know one single thing about it. Okay, so it's like, well, where do you start with this problem? So to explain sort of how we started to go about asking the question, how does this process work? Let me remind you, so this was our question, what makes these cells move and how do they do it? I'm going to remind you that your body is made of cells, as we've just been talking about, trillions of them. Each cell has a nucleus with chromosomes. The chromosomes are made of DNA. And you have about 23,000 genes that are living in these chromosomes. And so the DNA, of course, is a chemical code, and it codes for mRNA, and thanks to COVID vaccines, absolutely everyone knows what mRNA is. That's great. mRNA has the job, again, of coding for another type of molecule, a protein, as shown here. So for example, here is what the COVID spike protein looks like. But the thing is that each of your 23,000 genes codes for a different protein that has a different shape that serves a different function inside of our cells. So let's refine our question. Which genes and proteins control the migration of these cells called border cells? That was the question I wanted to address. And so how do we go about asking that question when you don't even know anything? At that point, we didn't have the genome or anything. And so what we would do is we would go in and we would break the genes one at a time, knock them out, as we would say. We'd go in and mutate the genes one at a time and see which ones, if any, were required for this. So if we break the genes one at a time, which genes, when broken, prevent my border cells from migrating? Now, there are about 13,000 genes that flies have, and so to go in and break them one at a time is actually quite a lot of effort, and so we do this in teams. And so this was the team at the time. We generated 10,000 different mutant fly strains, and I got one that had a border cell migration mutant. This was very exciting. Okay, so here normally the border cells migrate and they're in between the nerve cells here, and now in this particular mutant line, where one gene is broken, these cells can't migrate. Now this was great because it showed that you could find such genes. It allowed me to publish a research article, and I even got a job. So that was really good. So my first job, I was a faculty member at the Johns Hopkins School of Medicine. Okay, so, but now I had a problem because now I was sitting all by myself in my empty office, and how in the world was I going to get gene number 2? Okay, so fortunately for me, by coincidence, at that very moment, a journal article was published by Anne Ridley and Alan Hall, 2 British scientists. And what they showed was that one of our 23,000 human genes that encodes a protein called RAC, that if they tweaked this protein to make it active, that this had a remarkable effect on cells. And the effect that it had is not going to show up. Let's see, we were going to be able to play this movie if it didn't show up on here. I don't know if that's still possible, but anyway, what happens to these cells when they have an active form of RAC is that the edge of the cell gets really dynamic and starts like ruffling all over the place, and that's what these things are, they're ruffles. And so we didn't know anything about how cells move, but these cells got really dynamic, so I thought maybe that has something to do with cell movement. And so I wanted to ask the question, is RAC required for cells to move? And in particular, is RAC required for border cells to move? Now, RAC is a really ancient protein in evolution, the last common ancestor between plants and ourselves. Okay, so like the first eukaryotic cell had RAC. So flies have RAC. And so the experiment was to block the activity of RAC in border cells and see what happens. And this was the experiment that my very first graduate student, Anne-Marie Murphy, did. And lo and behold, we had gene number 2. So here, the border cells normally migrate from here to here and complete their migration. And in this RAC, when RAC activity was blocked, these cells could not budge. They couldn't move at all. So this was extremely exciting. Now, along the way, a postdoctoral fellow in my lab, Mohit Prasad, solved the problem that everybody working on this system had for many, many years, which was that you couldn't culture these egg chambers outside of the fruit fly. And until you could culture them outside of the fruit fly, you couldn't watch this happen live. So we were inferring that the cells were moving because they seemed to be in different places in different developmental stages, but here we could now actually watch them move. And this enabled many exciting kinds of experiments, including the one I'm going to show you here, that allowed us to probe the function of RAC in cell motility in greater depth. So this was a collaboration with a chemist, Klaus Hahn, and carried out by a postdoc in my lab, Xiaobo Wang. And what they did was very clever. They made a light activatable form of RAC. So they took, plants grow towards light, right? And they have proteins that allow them to sense light and do something different in response to light. And so what Klaus Hahn and his postdoc did was figure out how to capture that function of responding to light and put it onto RAC, so that now we had a version of RAC that was only active when we shine light on it. And they expressed that. We put that version of RAC into our border cells and then used a laser on the microscope to just activate RAC in one place. And wherever we activated RAC, the cells would extend out, and then the whole cluster of cells would migrate in that direction. And that would happen in this example, but that would also happen if we told the cells to go in completely the opposite direction to the one they normally move in. So it's like a video game. You can steer these cells around in a real tissue with light. So I jumped up and down when that happened. And so, so this led us to this idea, and I should say that RAC is now, my first student's experiment to show that RAC was required for border cells to migrate was really the first time that the function of this protein in cell migration was established, but now we know it's almost universal regulator of cell motility. Cancer cells, immune cells, embryonic cells, they all require RAC to move. And so we and others in the field, kind of got the idea then that when you had a little bit of RAC on one side of the cell, that told the cell which direction to move in. And this is usually in response to some sort of external stimulus. Okay, in the case of the border cells, the egg cell is emitting a beacon saying, come here, and it's activating RAC on that side of the cells, and the cells are migrating in that direction. Okay, so this was all well and good, all fitting together really well. But then we had a real puzzle, and that is, if we now took the super active form of RAC that Ann Ridley and Alan Hall had put into their cell, which made it ruffle around the edges, and we put that active form of RAC just in our border cells and nowhere else in this tissue, it killed the whole egg chamber. So if we put active RAC in this little group of cells, the whole egg chamber died. Now that didn't make any sense to us because this was kind of the same experiment that that Ann and Alan were doing in this fibroblast. They were putting active RAC in there and it didn't die. It ruffled and danced and stuff, And so it was like, so our cells shouldn't be dying, but moreover, I can intentionally go in and kill these cells, and I did. And it didn't perturb the development of the rest of this tissue. So what is going on? How is RAC activity in this little group of cells killing this whole tissue? Well, that was a 25-year-old cold case until recently. So we got a clue, again, from somebody else's work, in this case, Kim McCall at Boston University. Now, Kim was studying a later stage of development of the Drosophila egg. And I told you that these nurse cells, they donate all their contents to the oocyte, or most of their contents to the oocyte, actually. So the oocyte can grow, the egg cell can grow really big, and then the nurse cells shrink. And then they're not needed anymore, and what happens is that the cells that surround them, these follicle cells that surround the nurse cells over here, they actually eat them and remove them. Now, it's not uncommon for one cell to eat another, particularly if a cell is dying. We have cells that are professional eaters. They're called macrophages, which means big eater. And they go around our bodies and they vacuum up. They vacuum up lots of things like a virus-infected cell or a bacteria or a dead cell or other things, and sometimes even living cells. So our red blood cells, we turn over billions of them all the time. Those get turned over by being eaten alive. And what Kim showed that was kind of shocking was that these cells are getting eaten alive. So if you block the eating process, if you block engulfment of the nerve cells by the follicle cells surrounding them, the nerve cells don't die. They just hang out, they just stay there. So this meant that these follicle cells were eating the nerve cells alive. And so what, you say, has this got to do with anything we've been talking about? And so like I said, macrophages, or another word for a cell that eats is a phagocyte, they're cells that vacuum up debris and these kinds of debris. And so there are 150 proteins or so that are involved in the process of a cell eating another cell. But one of them is RAC, okay? So RAC causes this change in shape right there where it's gonna surround and eat this particle. And so we thought, well, could it be that border cells with this, hyperactive form of RAC are like mistakenly eating the nerve cells too early in development, and this is causing the death of the egg chamber? And so we decided to test that idea by eliminating this one receptor protein that we know is required from Kim's work, we know is required for the follicle cells to recognize the nerve cells as food. Okay, so this protein sticks out from the surface of the follicle cell and it allows it to tell that the nerve cells are food. And if we block that protein, our egg chamber that's expressing hyperactive RAC is now alive. Okay, so we've just removed that receptor protein and now the follicle cells don't recognize the germ cells as food anymore. And even though the border cells still have active RAC, they're not killing the germline. So that was pretty weird. But if we look at high magnification here, and I'm going to show you, the cells are still expressing hyperactive RAC, and now they're actually eating their neighbors. Okay, so normally the border cells actually live in harmony with these two cells here called polar cells. In fact, the border cells scoop them up and carry them. And that's what they normally do. But now, with hyperactive RAC activity, they're on a rampage and they can't see the germ cell, they can't see the nerve cells anymore, they don't recognize them as food, so they eat the polar cells instead. So here you can see the cell is completely engulfed. Uh-oh, I just saw a flicker. Well, anyway. Okay, so now we can come back to this idea that, well, a sort of local RAC signal on one side of a cell tells the cell to move in that direction. But then once it gets a really high and sustained and persistent signal, apparently it tells the cell to eat. Now that makes a certain amount of sense. If you think about ancient evolution, evolutionary time, we're talking about a single-celled organism, right, that has RAC. And what do those usually migrate towards? Well, they migrate towards food. And we've known for a long time that the leading edge of a migrating cell molecularly, is very similar to that phagocytic cup, the change in shape that the cell undergoes to eat something. So it kind of makes a certain amount of sense that the same signal, but just turned on stronger, would make the difference between moving towards something and eating it. Okay, so that was really exciting for us because we had solved our 25-year-old cold case and uncovered some, pretty interesting biology of cell, what it takes to make one cell eat another cell alive. And I have to say that in my field, in cell biology in general, we knew a lot about RAC, we knew a lot about phagocytosis, but everybody is surprised by this result, that just activating this one protein to a high level makes one cell eat another cell alive. But this would have remained a fairly niche finding in the Drosophila ovary field had it not been that about the same time, a paper was published showing that human patients are walking around with a mutation that hyperactivates a form of RAC in our bodies called RAC2. And these patients suffer from recurrent infections. So this is the shape of the RAC2 protein, and this is the mutation. And there were 3 unrelated people who went to the NIH because they had the National Institutes of Health because they were having recurrent infections and nobody understood why. They had their genome sequenced, and all 3 unrelated people had the exact same mutation. that changed the conformation, changed the shape of this RAC protein. So mom and dad were unaffected and they did not have the mutation, but the unfortunate child got this mutation and then had this problem. So, and this was published in this journal article, and we found that. And it turns out that the human patients have this activating mutation in RAC2, and they're immunodeficient, which is why they keep getting infections. And they're immunodeficient because they lack especially T cells. So this is data from that paper that I showed you, not our data. This is the normal number of different types of T cells in the body, and the people with the RAC mutation have almost none. So that's a very vulnerable situation. That's a very severe immunodeficiency. And so the human, in addition, so I should tell you that RAC2, we have 3 RAC genes. RAC is important enough that we have 3 copies of it. We have 3 different RAC genes. RAC1 is in every cell in your body. And if you had an activating mutation in it, you probably wouldn't get through embryogenesis. RAC2 is only in the cells of the blood. Okay, so the white blood cells, all the different kinds, the B cells, the T cells, and other types of lymphocytes. So you can get through embryogenesis and you can survive, and be born, but now susceptibility to infections. And it turns out that human patients with this activating mutation in RAC2, in addition to having too few T cells, they have hyperactive phagocytes, those cells that eat. And here's data from that paper again, and you can see here's a normal cell called, a particular white blood cell called a neutrophil, and it's a cell that goes around and eats up bacteria and stuff like that. And here from the patients, these cells are engorged, okay? But at the end of the journal article, this is what we knew. The human patients have an activating mutation in RAC2. They're immunodeficient because they lack T cells. But the medical mystery was, why are the T cells disappearing? RAC activity is actually kind of good for T cells. If you just T cells need a little bit of active RAC to be alive at all. If you take RAC away from them, they die. But didn't make sense why the T cells were disappearing. But based on our Drosophila work, based on our fruit fly ovary study, we wondered, could the patient's hyperactive phagocytes, these cells that eat, could they be eating the T cells alive in these patients and making the T cells disappear and making the patients immunodeficient? So to test that, a postdoc in my lab, Abhinav Mishra, he took some human macrophage-like cells and mixed them together with some living human T cells. And what he found was that the macrophage gobbled up lots of T cells, and he could quantify this big increase in how much eating went on if they had activated RAC versus just normal RAC. And so that was, again, exciting. And then a graduate student in the lab, Mel Rodriguez, she tested the same kind of idea using macrophages taken straight out of a mouse that had been engineered to have the same mutation as the human patients. And the mouse, which was engineered to have that same mutation, had all of exactly the same problems that the people have: depressed T cells, hyperactive, phagocytes, and so on. And she found, sure enough, a big increase in engulfment of T cells by macrophages that have this hyperactive form of RAC. So that was very cool for us. We had discovered something a little bit weird in a fly ovary, and now it was helping us to understand what was going on in these human patients with an immunodeficiency. So we felt like we had explained this medical mystery. But it got more interesting because we realized we might be able to harness this activity to treat cancer. So many people have heard at this point of CAR T therapy, I think. And the basic idea here is that you have a cancer patient, you extract T cells from them, you engineer them in the lab to make a chimeric antigen receptor, or that's what the CAR stands for. And what that does is specifically make the T cells bind attached to the tumor cells, tightly. And then you take those cells and put them back into the cancer patient, and now this has worked really, really well in a subset of patients who have a subset of cancers, like B-cell lymphoma, in some cases of melanoma. But the problem is that there are many limitations to this CAR T therapy. It is just an empirical finding that about 2/3 of the time the T cells can't even get into tumors, especially solid tumors, breast cancer, colon cancer, the more common cancers. Sometimes the T cells just pile up on the outside of the tumor and they can't get in. Macrophages are really good at wriggling their way into every nook and cranny, and tumors are packed with macrophages, normally. Sometimes the CAR T cells may not attach to every single tumor cell, and some escape, and that's because not every tumor cell is exactly the same as every other one, so they don't all have the thing that the CAR receptor is binding to. CAR T cells in general, and CAR T cells in particular, can enter a state called exhaustion, where they're not very functional. We can relate. And then CAR T cells can cause collateral damage by causing cytokine storms. And so there's a number of limitations, and therefore there's a newer cousin. So there's like 300 biotech companies around the CAR T kind of technology, but there's a newer cousin to CAR T, and it's called CAR-P or CAR-M, CAR-P for phagocyte or M for macrophage. I've been using those two words. Those are both mean a cell that eats. And so there's some potential advantages to this sort of CAR-P or CAR-M therapies, but the idea is the same. We get a molecule on the surface of this cell. We engineer the cell in the lab so that it makes this receptor protein on the surface that's going to bind to a target that might be a tumor cell. And then the idea behind the CAR-M is that one thing that'll happen is it'll engulf the cell and just physically remove it. Another thing that'll happen is it'll initiate an inflammatory signal to activate the rest of the immune system. It'll chew up and digest the tumor cells and spit out some of the peptides from that tumor cell, and this triggers the T cells in the body to activate a broader based immune response to it. And so the hope is that there'll be a multi-pronged attack on the tumor, both by physically engulfing it and by activating other cells to attack it. I had never heard of this CAR-M thing, but we had a candidate come through for assistant professor position at UC Santa Barbara, and her name is Megan Morrissey, and she was working with Ron Vale at UCSF, and this was their idea. They wanted to make some chimeric antigen receptors and trigger phagocytosis. And during her job talk, she showed some movies like this. So here's a macrophage encountering a B-cell lymphoma cancer cell, but it has not been engineered to have that receptor on its surface. And this is a nice little movie, and you can see that the cells interact, but you don't get engulfment. These are Megan's movies. And then here's a macrophage that does have the CAR receptor on it. And now you can see that sometimes the macrophage will eat one of the tumor cells. So that's good, but at the end of the talk, it was clear that it would be better if you could get them to eat more. We're like, we know how to make things eat more. So this was great. She was our top candidate. We made her an offer. She had other offers, but she accepted ours, and it was March of 2020. And everything shut down, and we couldn't do anything for a really long time. But our idea was, what we wanted to try was add to the chimeric antigen receptor, add active RAC, and say, can these macrophages now eat more tumor cells? And finally, we were able to do the experiment. And this is what we saw. Our macrophage was zipping around, gobbling up tumor cells. So that was, yeah, I know, yes, yay, I know, clap for the macrophage, love it. I'll show you again, it's really fun to watch. Yeah, so this was really exciting to us. And one of the things that was really exciting to us is what's shown here, and that is that the RAC by itself didn't do anything. So it wasn't like you were going to program these macrophages and they were just going to run around and eat anything. RAC by itself didn't do anything. It was the RAC plus the receptor that amped up the specific eating of the intended target. So that was pretty exciting. So we filed a patent on this, and it was licensed by this biotechnology company, Inceptor Bio, and they've been working with us to try to advance this technology towards a clinical application. We're excited because in the next few weeks, we're going to get our first human patient samples and try it all out on that. And so, okay, so we call this RAC-enhanced CAR therapy, or race CAR. And so this was a 25-year-old cold case solved in Drosophila, a mysterious human disease phenotype explained, and a new approach to cancer therapy that is very much in progress. Okay, so, but there are still limitations to this approach, and we would like to be able to democratize cellular immunotherapies. They're incredibly expensive. They cost $500,000 per patient. So the accessibility the cost is a problem. The time it takes to extract the cells, amplify them, engineer them, put them back, this is complex, it's expensive, and this limits access. So can we democratize it? And so this is what we really want to be able to do, and we think that the kind of technology that was used in the COVID vaccines might be able to help us do that. So the idea is, instead of taking the cells out of the body and engineering them to express a CAR and active RAC, why don't we make a lipid nanoparticle like a COVID vaccine that contains the RNA encoding the CAR and the RAC? And then it would be an off-the-shelf thing. You don't make a new one for every patient. You could use the same one on every breast cancer patient or ovarian cancer patient, infuse these lipid nanoparticles in, and now inside the patient's body, the macrophage is first going to eat the nanoparticle, then it's going to make protein out of that mRNA that's encoding the CAR receptor and the RAC, and now that's going to make it want to eat the target tumor. Okay, and so this is basically just in vivo engineering in the body instead of engineering outside of the body. So we don't have to take the cells out anymore. We don't have to engineer them and put them back. We can take this off-the-shelf medicine and give it to people. So this is very much just a concept, something we have written down that we think is very reasonable, plausible, maybe. And if we could do it, we know that you can do this at a scale that makes it affordable to give to a lot of people. And you could start thinking about treating things other than cancer with it. So anything you wanted to vacuum up that you didn't want in your body, maybe if you could make a receptor to it, you could program your macrophages to go eat it up and get rid of it. So that's the future that we envision for this technology. Okay, so that was one of my stories about how we went from studying border cells in the fruit fly ovary to what we think is a pretty exciting approach to improving cancer immunotherapy. And now I want to tell you, I mean, you could say, well, you got pretty lucky there, and I would agree with you that it was quite the coincidence that Megan Morrissey came and interviewed, and this company was interested in this technology at the time we had. There were a lot of lucky things that happened there. But, lightning has actually struck twice for us, which is pretty exciting as well. And so this is led from another project that started in the fly ovary to what we think is a possible gene therapy for a variety of neurodegenerative diseases. So retinitis pigmentosa is a genetic blinding disease. This is the retina from a retinitis pigmentosa patient. And what happens is that the photoreceptor cells, the cells that sense light in the eye, die. And they die because of a mutation in the light-sensing protein rhodopsin. So this is the protein in your retina that senses light and allows you to respond to light. These people have one copy of the rhodopsin gene that's perfectly fine and one copy of the rhodopsin gene that has a mutation that causes the protein to fold in the wrong way. So it doesn't make this very organized shape that is important for its function, but rather it folds up in a very disorganized way. And the problem with that is that misfolded proteins can clump up and then clog the cellular plumbing. Okay, so when you have a misfolded protein, you have a lot of it, and photoreceptor cells make huge amounts of rhodopsin. You actually replenish your rhodopsin like every morning before you wake up. You replenish the rhodopsin in your eye, so you make lots of this protein. And if you make a lot of misfolded protein, the cells starts to clog up. And this is because cells literally have a garbage disposal, okay, for misfolded proteins. And it looks like this, like it actually looks like a garbage can, kind of. And the misfolded proteins get put into this garbage can, and then they get chopped up into little pieces and recycled. So cells are pretty good at doing things like that. And the problem is that because of this mutation and because of the large amounts of rhodopsin protein that are made, the cell just can't handle the load on the garbage disposal. It clogs up, and the cell ends up dying. This happens, these, right, yeah, over, right. Now, it turns out that misfolded proteins cause more than just retinitis pigmentosa. They also, what happens is our garbage disposals in our cells tend to get clogged and not work as well over time with age. And so misfolded proteins can accumulate and drive age-related neurodegenerative diseases like Alzheimer's and Parkinson's and ALS and others. So this is a fairly common phenomenon. Right, here are some of the diseases, and here are some of the proteins that are misfolded that associate with these particular diseases. So misfolded proteins can clog the cellular plumbing and cause degenerative diseases in flies too. And so here again is the human retina, that's the healthy retina, and the one from a retinitis pigmentosa patient. And here's a healthy fly retina. And then if we express the same mutant form of rhodopsin in the fly that the patients have, we get retinal degeneration and blindness in the fly as well. So what we have found is a gene called ZIP7 that when we add this gene to the fly eye, it actually prevents the blindness due to rhodopsin. So these flies are expressing the same mutant rhodopsin that this one is, but we've added to it another gene called ZIP7, and this fly now has a perfect morphology in the eye and can see perfectly fine. So what is ZIP7? Well, I could tell you a long story like I told you about RAC, about how we sort of figured this out. It's a fun story, but it's getting late, and so I won't bother you with it. But the bottom line is that what ZIP7 does is make the garbage disposal work better, okay? So it just means that it can handle more volume. And so that was pretty cool because nobody in this field had any inkling that would be the case. But what I want to leave you with is the question, like, how did we find ZIP7? And we weren't even studying this garbage disposal or misfolded proteins or anything. We were studying border cell migration, right? But we found ZIP7 by screening for mutants with defective border cell migration, and this is one of the mutants that we had. It turns out that this protein encodes, or the protein encoded by the ZIP7 gene, again, this is deeply conserved in evolution. Plants have ZIP7, we have ZIP7, flies have ZIP7, can't live without your ZIP7. And yeah, we were able to figure out based on the work in border cells that this is the function of ZIP7. So, and that we can rescue the blindness in the fly, and so it is a candidate for a gene therapy. The only gene therapy that's approved for use in humans is in the eye. So the eye is a reasonable target for gene therapy. But well, I told you that many neurodegenerative diseases are actually caused by misfolded proteins clogging up the works in one kind of cell or another. And so how good is ZIP7? Can it rescue any of these other protein folding degenerative diseases? And the interesting thing is that you can express any one of these misfolded proteins that is known to cause neurodegenerative disease in a human. You can express it in the fly eye, and it will cause the fly photoreceptor cells to die. So we can test out our ZIP7 in the fly first and say, is it a good candidate for any of these others? And then we can if the answer is yes, we can take it to more realistic models of human disease, like human cells. But we start with the fly, and what we found, we've only tested 6 so far. There's like 300 misfolded proteins that cause degenerative diseases that you can express in the fly eye and kill the photoreceptors in the fly eye. But we've only tested 6 so far, but it's worked in 3 of them, which is pretty cool, including A-beta 42, which is associated with amyloid plaques and Alzheimer's disease. 100% of the fly eyes are deformed and non-functional when we express A-beta 42 in the photoreceptors, and this is reduced to about 40% when we also add extra ZIP7 to those cells. And this is a familial ALS gene, VAP33. 100% of the fly eyes are abnormal. The photoreceptors die if we express this aggregation-prone protein, and we can rescue that to about 35% when we add to that ZIP7. So we're pretty excited about these findings and interested to see just how many of these misfolded proteins we can overcome and rescue the cells from. So ZIP7 then, overexpression is potentially a new gene therapy for retinitis pigmentosa and possibly other neurodegenerative diseases. So I just want to really leave you with 3 thoughts. And that is, first of all, that curiosity can lead to cures. That curiosity, just pure curiosity-driven research. How do the border cells migrate? That just looks so cool. I want to really understand that. Can take us in directions we really don't anticipate. And then I want to say that this research is an international team sport, that this is my lab at one particular moment in time. We have people from India and China and South America and Africa and Eastern Europe and Western Europe and all across the United States and from all kinds of varied backgrounds. And it's literally this melting pot where all kinds of creativity and ideas just turn around, and it's an incredibly supportive and collaborative group of people who cheer each other on every day. And so it's really important that we continue to have free flowing of people in science across country borders and all of these things, because it really is an international team sport. And then stay tuned for race car and zip gene therapies coming your way soon, I hope. And this is the beautiful environment that inspires us every day. That's literally the view out the window of my office. And I have funders that I need to acknowledge, including the National Institutes of Health, General Medical Sciences, Institute of Aging, and Inceptor Bio. And I will stop there and take your questions.